here. Good. Eliana Crespo is here. Eliza. Okay. Emily. Leonard Garcia is here. Janice. No Janice. Okay. Kimberly Gruber is here. Good. Jimena Hernandez. Okay. Rafael. Good. Tatiana Lopez is here. Caitlin Morales. Good. Gabriella. Right. Eric is here. Good. Okay. Samantha Orr. Good. Jetta. Where's Jetta? Okay. Jacob. No Jacob. Oh.
He's coming tomorrow.
Oh, he's coming on. Okay, yeah. That's right. Okay. Tali. Okay. Desiree, sorry. Yeah, okay. Mustafa is here. Kate Santana. Good. Maritza Vera.
Yeah.
Okay. What? You have the same name? Okay. No. Kiana Williams. Right on. And Janet. Where's Janet? There we go. Okay. Ariana. Okay. Terrence. Andrew is here. Rossi. Good. Vivian.
Yeah.
Okay. Tyler.
Here.
Good. Michaela Guerrero. Nancy Gutierrez. Oh, right in the back. Talia Jenkins. Arthur. Brenda. Tamiko. Okay. I think did you pick up your information sheet to correct? Okay. Santali Ortiz. Okay. Here or no? Okay, so we got I've got Titali Rivera. Okay. And I've got, okay, good. Anna. There we go. Josh. Okay. Joanna. Good. Manuel. Good. Annabelle. Maritza Rivera. Here. Okay. Melissa San Pedro. Alexandra. Melissa Torres. Okay. Grace is here. Good. Okay. Julissa. All right. Last. Haley. Victoria. Good. Sydney. Ebony. Good. Madeline. Chloe. Okay. Michelle Dang is here. Good. Sierra. Right on. With the new iPad cover. Alaya. Elia Guzman. Going once. Okay, Catherine. Jada. Good. Jesse. Good. Victor. Hugo. Soleil. Where? Okay. Is that being turned in?
Yes.
Oh, okay. It's not complete. Okay, Alyssa. Anaya. Good. Justice. Good. Raul. Good. James. Good. Ivan. Let's see Ivan today. Paulette.
Here. Hi.
Oh, right in
Sorry.
front of me. Okay. Anna. Okay, good. And Minahal. All right, good. Cool.
What is this? Two.
All right, let's get started today. All right. So you should probably get by your the people that you did your cards with and another group, because you want to take out your cards and take a look at them now. So I'm going to give you a minute to get seated by whoever you did your cards with and another group. Okay, and get out your cards. Yeah. Get it? Yeah. Oh, I just, yeah. You know you sort of You just gotta think. Okay, have you got with a group, another group you did your cards with? No, it gets stolen routinely, so yeah. Okay, now that you've gotten your group together and you're with by another group with cards, okay? Cards and cards. Compare the cards. What's your cards?
Yeah, that one was easier. Because the information sheet kind of listed like that they put them together. Yeah. And I was like, what is that shit? That one, yeah, that one has the numbers. Yeah, like that one I heard like the other day and I was like, I have no idea. I know, at first I was like, I thought like five were all like, yes. But then yeah, like that, I was like, oh, okay, then. They're just like similar. Yeah. Like, I think it's the right one or something.
To get something, you just get like briefly mentioned.
My favorite was
With, like,
yeah, my favorite was
so we'll be there. Like what they share.
So for my part, I kind of, I'm not the most tidy individual, so I just kind of like random. For like the these numbers that have like types, I like read it. Or like specifically like symptoms, so it's easier to I still have to say something. I have like four numbers, just because I didn't know numbers, but I just kind of did everything that was like titled to or like anything that was
Yes.
and I can use our code later.
This number. 0700.
I found these little envelopes, and I asked her because I was wondering if I can have them together, like and she said, I can have like this, and I went, everything is colored.
You know what I did? It was like the like oh my like I know what it is now, but like the first time I was beginning to so but this is number one and then it's two sides. And then I left the page blank and you send it to like you know. That's number two. The same thing is review.
How's it going? How's your cards? Okay. Where's your cards? Bring them. No, you gotta bring them. It's not every day. Every day. Yeah. What?
Oh, because I found it.
Oh yeah. How's it working for you?
So I think it's I work for me because I would like to have another and then I did like I did number one and then I left the patient
Oh, in case you need to add something. Okay, that's cool.
but I'm not sure if I could we did one
Oh, are you good?
Oh my God. And you tried messaging him and he's not. Okay. Is it okay? Help each other out? Yeah. Okay. All right. Okay. So,
but you guys already
a couple of things
write it down.
to be aware of is that what your cards, you gotta bring them every meeting, every single meeting, because there are times as we get talking about systems and tests and things like that, I'll say, okay, do you have this on your card? Okay, and if you're like, oh, I left him home, or you know, so by now you should have all 10 done. Okay, and some of you printed it right out from the sheet or reorganized it, but then you should have one disease per card. And next time we come to class at 9:35, the quiz will start. The quiz, you can use your cards. That's it. You'll get the questions up here. Okay, the questions will be on a PowerPoint. Okay, and you'll have about a minute to answer. So you've got to know your cards very well. Okay, no printouts, no thing, oh, I've got it on my iPad. No, you've got to have physical cards. Okay, so yeah, any questions about that? Yeah.
What chapters is the quiz going to be on?
It's just going to be on the cards. Yeah, it's just going to be on the cards. Yeah.
It's not on the scientists yet, too?
No, I'm not going to put any, I might put one question about a scientist, maybe, but I think right now I want to make sure that you've got your cards down. Yes? Yes, you can. Yeah, you can put, if that's, you know, if that will work best for you, that's fine. Yeah?
Does the quiz require the Scantron?
No, I'll give you an answer page for it. Yeah, so you just have to come in, make sure you're here on time. I'll hand out the answer pages. We'll start the quiz. Okay? Does that make sense?
Yes.
Okay, yeah. So always, so just add it to your bag. When you come into micro, bring those cards, okay? All right, are you learning from them? Like you're learning about more about Staph aureus than you ever wanted to know, right? And for our dental hygienists, you're learning about all the things that cause dental caries, right? Which are cavities. All right, cool. Okay, good. Anything else? Any other questions or anything? Did it help to see how other people are doing their cards? Okay, this just kind of makes you think a little more. Yeah.
So for number 1 through 5, there's more than like 5 actual diseases listed. When we break them up, how should we have sectioned them?
Okay, so the first one is triple S, and then it gets into the boils and the furuncles and that you could put that on one card, because all it is how they erupt is a little different, right? Okay, so that will compact that one. Okay, no problem. All right, any other questions? Okay, you good? How's it going so far? It's a lot, yeah. This is 50B on steroids, totally. Okay, 50B on steroids, without a doubt. Okay, but this is a capstone class, right? So this is your one and only NCLEX. I got to talk to a couple of micro students I had years ago that are working. One's a travel nurse. Talked to her over the weekend, and I said, okay, so out of micro, what did you use the most? And she said, oh my gosh, the cards. I was like, really? She goes, yeah, because they're embedded in my head. So I look at something and I know. So she's a charge nurse at this hospital in Michigan right now and loving it, okay? She lives out of her camper and makes a fortune and then goes and plays for like 2 months. Like, comes back down and goes surfing and has a life. And I said, well, what was the hardest thing about the class? And she's like, just the volume. It's just the volume. It's not hard stuff. It's just a lot. Yeah, and you have to really know it. So, yeah, but you're all smart. You've made it this far, right? Yeah, you've made it this far. You can do it. I told you the first day, thousands have come before you and thousands will come after you. So you can do it. It's just a matter of choices, right? Choices. Yeah. Do I study or do I do this? Do I this or this? And I really feel, I don't know, kind of, society says, oh, you can do everything and anything you want. Yeah, just not all at the same time, right? It's kind of misleading, you know? Oh, you can do anything. Well, it depends, you know? Yeah, so just kind of think about that. All right, so let's get into epidemiology. Wow, what a crazy weekend for infectious disease, wasn't it? This has been a crazy time. We've got 1 dead from measles, probably more to come. We've got that epidemic or some unknown disease in the Congo that we still can't find that has killed almost 100 now, over 100 now. So yeah, lots of things going on in terms of diseases. So let's kind of start where we left off last time, okay? And this was the last thing we looked at, which was sources of infection and talking about animal reservoirs. And we started talking about what happened in the Congo. Obviously, these folks got sick from what animal? The bat, yeah, don't lick bats. So I have 2 rules in life. Number 1, stay hydrated, and number 2, don't lick a bat. Don't eat a bat. It seems like they carry so much that they're mammals, so they're gonna carry a lot of infections that we're gonna be susceptible to, okay? Okay, so we've talked about zoonoses, disease transmission naturally or directly or indirectly from an infected animal. Okay, one of the swine flu, H1N1 is a good example. Bird flu, H5N1, that's another good example. The mystery illness in the Congo came from an animal. Okay, so presumably a bat, since that's what those children ate. Okay, so gave a lot of examples here of zoonoses. Okay, about zoonotic disease. Okay, so we've got a bunch here, rabies. Oh my goodness, yeah. Do you remember that teacher up in Fresno that found a bat in her classroom. This was over the end of last semester. She had just started, she came into her classroom, I guess, over the weekend or after the weekend, and there was a dead bat, so she picked it up and threw it away. And it bit her. She died of rabies, but the incubation time for rabies is so long, it's months, and so she had forgotten that it had happened, and so by the time they diagnosed it, it was too late. So yeah, these things happen like all the time. So yeah, don't touch the bats. Okay. All right, you're getting the idea. I love tables. I love tables, but do you have to memorize the table? No. But should you be familiar with it? Absolutely. Okay. So here's some if you notice, a lot of the zoonotic infections are viral. Okay. And then there's some bacteria ones that come, through ticks and birds. I have a student in Bio 1 right now. We were doing microscopes this last week, and he said, I can't see through my microscope. And I said, okay. And I looked at it, and he said, well, I'm sort of blind in my one eye. And I was like, okay, that would make sense. And he said, well, then he said, well, my mom took care of the kitty litter when she was pregnant with me, and I got toxoplasmosis. And I was like, you're kidding. I have not heard of a case like that in ever, not in this country. And he said, yeah, so yeah, I'm blind in that one eye, and I can't see too well. And he says, with those infections, they're always low key. I said, yeah, they're always in you. And he says, when I get really stressed out or tired, then I have all these symptoms again because the microbes come back. So yeah, it's no joke, the things that we pick up from animals, and especially, the susceptible pregnant ladies. So, all right. Yeah, more. Lots more. See, here's toxoplasmosis. Okay. Yeah. So, how did all these transmit? Well, oftentimes it's airborne or vectors, which means it's an insect that carries it. Okay, close proximity to the oral fecal route of animals, consuming contaminated food, okay, or bites, okay? So, yeah. There's a difference between a definitive host and a dead-end host. Once that poor teacher got rabies, she couldn't pass it on. She's a dead-end host. Once you get, these microbes, so far for H5N1, humans are a dead-end host. That will probably change as it mutates, but for now it's a dead-end host. So, okay, so some more examples of zoonoses, okay? Usually you don't want doggies swimming in swimming pools because unless your pool is perfectly taken care of, they have leptospirosis. They can transmit leptospirosis from Leptospira spirochetes. Now look at the shape of this. Look at the shape of this. Wow. Okay, that tells you its function. Okay, it's got a spirochete shape, so you know it's going to do what in the tissue? It's going to burrow in. Yeah. Okay, and it can come and damage organs. So yeah, it's fun to have the doggies in the pool, but boy, you better have taken perfect care of that pool, or else you could get leptospirosis. Okay. All right. Let's talk about infection sources. There's non-living reservoirs of infection sources: soil, water, and food. And I'll give you some examples of what happens with soil, water, and food. Okay, you've got Clostridium tetani, which gives you tetanus, botulinum toxin, fungi that can cause ringworm. In the water, you can have Vibrio cholera, Salmonella, Giardia, and for food, Staphylococcus toxins, Salmonella, and a big one right now is Listeria. Listeria. Have you seen how many cases of listeria? There's been a ton of cases, and one of the causes is a protein drink that was given in long-term care facilities. And so, of course, you've got this susceptible population. You've got listeria that's contaminating. Of course, they passed away of their infections. So, yeah, it's been a bad year for listeria. So, yeah, I told you my cousin died of food poisoning right before the semester started. Yeah, he died of it. Him and his wife were hospitalized, and yeah, they were healthy. They didn't make it. Well, she made it. She's still recovering, but he died. Yeah, this is our food safety is a real problem in the United States in terms of, you know, we're getting lots of cross-contamination and stuff like that. It's not good. Okay, we've been introduced before to nosocomial infections. These are diseases that are acquired when? Hospital or health facility stay, like a SNF, right? Care facility, okay? Up to 10% of admitted patients may acquire an infection, okay? And it's sort of the intersection of all the microorganisms you have in the hospital, the chain of transmission, and then you've got this compromised host. You've got a patient in there that's compromised. That's the trifecta that leads to nosocomial infections. And again, what is the most common nosocomial infection? UTI, yeah. And what's usually the causative agent? E. coli, yeah, E. coli. Okay, so we sort of talked about this before. Yeah, most are UTIs, the other are respiratory. Some of the infections in surgical wounds, okay. So you have UTIs, respiratory infections, and surgical wounds, okay? Universal precautions. That's really what's gonna help this out, is universal precautions, okay? And what are universal precautions? This is the guideline established by the CDC. It was after the HIV/AIDS came on the scene. So it was the late 80s, early 90s, okay, to reduce the risk of disease transmission in a hospital and lab setting, okay? So if you're not familiar with universal precautions, set some time out to go look at what those universal precautions are. Okay, a lot of you are working in healthcare, so you know already, okay? These are some ways that we can reduce nosocomial infections. If we use universal precautions, the infection rate would be cut in half. So when you see a hospital-acquired infection, oftentimes it's because of the failure of universal precautions. So there's lots of different kinds of precautions. Do I expect that you know all of these kinds and all that? No. But again, it's a table. It's good reference, right? Okay, so there's enteric precautions, respiratory precautions. We're all real familiar with that right now. Strict isolation, reverse isolation. Okay, just so you're familiar with those, that's one of the ways that we can, is just by having these different levels of isolation. Okay, good so far? Okay, let's talk about how infection is spread, the transmission. Okay, when you talk about a communicable disease, that means that it is capable of spreading from a living host to another living host. Okay, non-communicable disease is picked up from the environment, but cannot be passed on to other things. So like things like Giardia. Well, actually you can pass that one on. Not very easily, but you can. But there's two kinds here of non-communicable, where you just pick it up but you can't transmit. Okay, like rabies. She got it from the bat, but she can't pass it on. Okay, and then you've got communicable diseases, and we spend a lot of time talking about these, those that are transferred from one living host to another. Okay, So we're going to talk about how these transmissions occur. Okay, and if you've got different colors, it helps with this because there's so many variations in how disease is transmitted. But you've got three general groups: contact transmission, vehicle transmission. I'm not talking about your truck or your car. You know, oh yeah, vehicle transmission. Oh yeah. Okay, vector transmission. Okay, so we're going to learn the different kinds here. Okay. So let's start off with contact. So if I was taking notes, I would put everything in one color so I can remember what contact transmission is. Okay? So we've got three things that fall under contact transmission. Okay? It requires, first of all, is direct contact. This is the easiest one to think about because in order to get this, what do you have to have? Direct contact, like with Mpox. Okay, if you read about Mpox, the clades that are out right now are, they say they're aerosol, but the cases we have been direct contact, okay? So there's two ways that you can get direct contact. Horizontal transmission, which means that you spread it to the people around you, okay? Or we have vertical transmission, which it's mom to fetus. So my student that had toxoplasmosis, what kind was it? Vertical, yes. So as you go through your cards and you get examples of things, I mean, we've just started with 10 diseases, we've got some more to do. But when you find, examples of, contact transmission that are unusual, make sure those go on your card, okay? All right, so any questions about direct contact? Okay, is this direct contact? No, okay. Now, direct droplet transmission, okay? We see this all the time because what happens? People what? Cough and they sneeze, all right? One of the things I think that the U.S. public health system failed the U.S. so severely is when COVID first started, and Fauci, when he was brought before the panel, said, well, we weren't sure about 6 feet. And I was watching and I thought, 6 feet? 6 feet's a magic number in public health. What is 6 feet for? That's as far, if you don't cover a sneeze, that's as far as your droplets go out, is 6 feet. Okay? So when he testified, oh, well, it was just an arbitrary number. No, it actually isn't. 6 feet is what we, if you blow a sneeze out at 200 miles an hour, it's actually more like 150 miles an hour. You're going to spray droplets for 6 feet. Okay? Now, which is bigger, a droplet or an aerosol particle? The droplet is bigger. Okay? So we're hoping that a lot of droplets fall to the floor, which makes the floor's, hygiene extremely critical. Aerosols are different, okay? You sneeze, you produce a lot of droplets like the picture shows, but you also produce a lot of aerosols. Aerosols are lighter, they're tiny. They can hang in the air for hours, okay? When that case of measles came through LAX last week, okay? One of the things we were talking about at dinner was, oh my gosh, what about everybody on that plane? Well, how is measles transmitted? It's aerosol and droplet. So did everybody on that plane for hours get exposed?
Yes.
Absolutely, yeah. What's the incubation time for measles? Up to 21 days. So people have got to, some people are having to isolate for up to 21 days, okay? All right, so we've got direct droplet transmission, and then we have indirect transmission. What is this? Requires this is my favorite F word, fomite, yep, fomite, non-living objects that can transfer disease from, so if I were to use, I don't like to touch it. If I had this, that would be a fomite. We talked about fomites, but that's an indirect transmission. Okay. So, have you got the 3 types of contact transmission so far? You good? Okay, let's move on to the second one, which is vehicle transmission. Vehicle transmission, a vehicle is a non-living carrier of a microbe from its reservoir to its host. Okay? So we have water, and the example is the oral fecal route. Okay? Giardia is a great example of that. Anybody know what the host for Giardia is usually? The host, the living host. What's a living host? Beavers. Yeah, they call it. Yeah, so beavers carry it. There are other hosts, but that's the most famous one. Okay, and so what happens? The animal poops in here. We drink the water. Ta-da. Okay. Aerosols, long distance droplets or dust. They can be vehicle transmission. All right. We just had big fires in LA. One of the things that we see after big fires in Southern California or the Central Valley is that we're going to see a lot of fungal lung infections show up. Okay, they call it valley fever, coccidioidomycosis. Okay, it's a infection where, because of the fire, it stirred up all these aerosols and dust. People breathe it in, it stays in the air because it's light, okay? And those fungal spores attach to the particles, and it's inhaled by people, and they get that fungal infection in their lungs. So, yeah. And then what is this? Food. Yeah, food. Okay. What are some of the outbreaks we've seen in food? E. coli, right? What? E. coli, yeah, totally E. coli. Lots of E. coli, especially here, yeah. Okay. Any question on vehicle transmission? Have you got those examples? Yes? Aerosols. Yeah, aerosols. Okay. All right, let's talk about vector transmission. Vectors are biological vectors that transmit disease from one host to another. Okay? So, the vector could be an insect, okay, or a rodent, like with hantavirus, it's mice, or it's birds, okay? So, these are all vectors, okay? And so that's why oftentimes if you work in a place with patients, they're really concerned about vector control. Why? Because these can directly cause infection, but look at this. You can also have mechanical transmission. What does that mean? You might not get bitten by this fly, but what does a fly like to hang around? Poo and food and things that don't smell good, and they get it on their body, and then they move to your food and leave it there. Okay? So that would be an example of a mechanical vector. Okay? Versus a biological vector when you get bit or something like that. Okay? Does that make sense? So you've got these two methods for vectors. Okay? All right, so what are the 3 we just talked about for transmission? Contact, vehicle, and vector. This would be a really nice matching section on the exam, okay? So to make sure that you know your ways that things transmit. And I just found this morning this really cool video that goes through and talks about these to practice. So I'll post that this afternoon. Okay, so there's direct and indirect, like using vehicles. Direct is contact, droplets, vertical, okay, and biological vectors. And then indirect is fomites, food and water and aerosols. And aerosol to down air, okay? So, yeah, so this is, so if, for example, if you got something like this, syphilis from a tattoo, these are very strategic lesions, aren't they? I mean, I'm looking at this, okay? So, this was back in 1877, okay? He infected his clients using saliva, which he used to moisten the pigments. He would sometimes spit on the completed tattoo and rub it with his hand or a dirty cloth. Yeah. Ooh. Yeah. What kind of transmission? Or what types of transmission? Talk with your study buddy. What kind of transmission would this be?
Yeah, right, direct contact. Direct contact at least with his hand.
Yeah, there's lots of contact, right?
And then the droplets, obviously.
Okay. What'd you come up with? Direct, okay. Droplet. What else? I would say indirect using fomites, right? Because he's using the
cloth.
the needle. He's licking. Yeah. Yeah. All sorts of problems, right? Whew. Okay. Good. All right. So, if we know that all these ways of transmitting microbes, how do we block that chain? First thing we want to do is isolation of a patient that has a communicable disease. Okay? This is a putative Ebola case. Okay? They got them all lit, so it is all isolated. Okay? This is a quarantine station. And where did Mary Mallon, Typhoid Mary, stay? North Island in New York. So yeah. Okay? Then quarantine. Oh, okay. Quarantine. This is where you separate healthy people that have been exposed to a communicable disease. Okay? Then we have immunizations. Okay? We've talked about immunizations. And then finally, vector control. And using these, we can cut down on a lot of transmission. Okay? So, all right. So, any questions before we go on to this section, which is we're talking about the disease process? Good? Okay. This provides an overview of what the disease process is, portal of entry, and then you have two things that can really affect if the microbe's able to get, into the host. It's entered the host, but can it adhere, and how many microbes, okay? And then you've got penetration or invasion of the host, defenses, damage to the host cells, and portal of exit. Now, just so you know, the portals of entry may not be the same as the portals of exit. It might be, like for respiratory things, that's often it, but it's not always the case, okay? So what we're gonna do is gonna go through each of these boxes one at a time. And we're gonna start with the portals of entry, okay? What's the first defense you have against any invasion? Skin, yeah. But there's always something to consider. There is no such thing as an intact epithelium. Look at your hands. Do you have any cuts on there? Do you have any, if you got your nails done, you absolutely got cuts all over the place. And you may not even see them, but they're microscopic. Do you have that in other places? Yes, of course. Okay. So skin is a really common portal of entry. The next is respiratory and digestive tracts. Then it's genital urinary tract, mucous membranes, and the placenta. Okay. All right? So you have to get the microbe through the right portal of entry. If you've ever noticed, you've never gotten pneumonia in your toe. Okay? Why? Because it doesn't have what it needs in order to cause infection. But, you know, you look at Klebsiella pneumoniae, it is common in the pharynx in the winter. In fact, we probably would if we swabbed, would test positive for that. But it's in the pharynx. In order to cause pneumonia, where does it have to be? In your lungs, okay? But do we get pneumonia of the pharynx? No. Where does it tend to cause infection? It has to get to the right place, okay? So, all right, so those are the common portals of entry. The placenta. Yeah. So, pathogens, speaking of the placenta, okay, remember this, TORCH. It's a great acronym to remember all of those diseases that can be passed from mother to child in utero. Toxoplasmosis, which we talked about this morning, syphilis, the other diseases, that's the O, syphilis, varicella, parvo-19, HIV, rubella, cytomegalovirus, and herpes simplex virus. Okay? Back in the day when my student's mom was pregnant, they didn't think about TORCH and educating the moms that, hey, you can get, don't be changing the kitty litter because of toxoplasmosis. And that's now we do. That's why we don't see too many cases of it anymore. Okay? All right. So let's talk about the disease process. So we're going to go back to here. Here's this, and now we have to think about how does this disease process happen, okay? There's a term called pathogenicity, and it's the ability to cause a disease in a host. And virulence tells you the degree of infection, okay? So whenever we've got an infectious agent, these are two things we want to know from the top. What's its pathogenicity and what's its virulence? How severe infection is it going to cause? And we have to consider all of these here. But let's give you some numbers so you can actually do some comparisons about this, okay? When we talk about pathogenicity, we talk about infectious dose. It's the minimum number of microbes that is needed to cause an infection. Okay? So, we call it ID50. Okay? ID is infectious dose, and this is what is known as pathogenicity. It's the number of cells required to cause 50% of healthy hosts to become infected. Okay? For measles, the infectious dose is 1 viral particle. Okay? There are some that you need hundreds of thousands of viral particles to out. Okay? We also have this term LD50, and you may have seen this with drugs. It stands for the same thing, lethal dose 50. It's how many will die. Okay? So how many will die, or the cell number that is the concentration that will kill 50% of healthy animals. Okay, so you see this with drugs, you also see it with microbes. Okay, so this gives you the pathogenicity, this gives you the virulence. So let's do a couple of examples down here so you can compare. An ID50 of Shigella is 200 cells, and for Salmonella, it's 10 to the 5 cells. Which is more infectious? Shigella. It only takes 200 bacteria cells to make you sick, but you need 10 to the 5th cells of Salmonella. So Shigella is much more, it's ID50, yeah, it's much more infectious, okay? LD50 of botulinum toxin is 0.000025 milligrams. Well, salmonella is 20 or 200 milligrams. Which is more virulent? Botulinum toxin. That LD50 is much lower, okay? It doesn't take much. Yeah?
What does the L mean again?
What does L mean? Lethal. Lethal isn't dead. Yeah.
What is P and B?
Okay, help them out. What is P?
Pathogen.
Pathogenicity. What is V?
Virulence.
Virulence. Yeah. Yeah. Okay. So here's another table that will kind of tell you. Here's the infectious, the ID50. Okay. Yeah. So norovirus, I know that's been going around like crazy. You need 18 viruses or less. That's it. No wonder this is so many people get it. So, yeah. Okay. All right. So, what other things? When we talk about pathogenicity, what are the factors that affect pathogenicity? Well, it's the number of microbes, it's the attachment, it's the avoidance of phagocytosis, invasion of the immune system, production of extracellular enzymes, production of endotoxins, production of exotoxins. Okay. So I know we go over these in lecture, but if you can try and get these filled out from chapter 13 before you come into lecture, then you can just kind of double check and see if your blanks are correct. Okay, and then it will be your second time through it, right? Okay, so all of these things, 1, 2, 3, 4, 5, 6, 7, affect the disease process, affect pathogenesis. We're going to go through them one by one now. Okay, so number 1 is the number of microbes. Okay, we talked about that. You could see some infections take 10 to the 5th microbes to infect. Some you just need 1 virus. Okay, this is why people were asked to cover their mouths and nose during respiratory infection. What are you trying to do? You're trying to reduce the number of microbes that are out there circulating. Okay, here's attachment. Okay, this is the fimbriae. Okay, glycocalyx. All right, let's look at fimbriae. These are really neat. Okay, see these little like hair-like things that are here? Those are fimbriae, and they're sticky, and they help the microbe to stick to the environment. Okay, those are the fimbriae. These are really common in STIs. Why? Because when the microbe gets to your genitourinary tract, it will stick to the walls using these fimbriae. Okay, glycocalyx. We're going to talk in detail in chapter 4 about a glycocalyx, but a glycocalyx is, what does glyco mean? Sugar. So glycocalyx is the sugar coating that is around the microbe that can make it sticky or make it slimy. Okay, so it sticks. Okay, and one of the things you'll see are capsules. Okay, and you can see here's a microbe and it's surrounded by this, the cell is tiny, but it's got this giant capsule around it that protect it so it doesn't get phagocytosed. Okay, we've got also called adhesins, which are that bind to the host receptor. Okay, we've got a picture of how that works here. On the microbe, you've got a ligand, and you've heard of ligands in chemistry, but you also have ligands in cells that help with cell signaling. And the microbe will bind to a particular receptor on the host cell, okay? All right. The other thing that we have that help microbes attach are biofilms. Biofilms help microbes attach to all sorts of things. Your teeth, and what is a biofilm? This right here, you can kind of see, actually this is okay, but you can see the microbes. When the microbes get together to make a biofilm, oftentimes it's more than one species that comes in, okay? And what they do is they secrete sugars, they secrete proteins, they secrete lipids that makes a protective film around the bacteria. And it makes it harder for the host because our immune cells aren't really good at getting rid of biofilms, okay? But we're gonna see more and more of biofilms as the semester goes on, because that's usually what causes the most common nosocomial infection is a biofilm, okay? Yeah, is that good? All right, what else do we have? We also have viral spikes, okay? Parasitic suckers. So some of the parasites will actually have a sucker to attach, like tapeworms. Okay, and then we also have suckers and scolex. Scolex is part of the tapeworm, and it's part of the head of the tapeworm. It's called the scolex. It's got some hooks that come in and a sucker to attach to the intestinal wall. So, okay, so we've done number 2, which is attachment. Lots of different mechanisms of attachment. Okay, and here's another table that kind of shows you, again, you're not going to have to remember the table, but it's just for your reference, so you have it, okay? Now, how do we avoid phagocytosis? Okay, first of all, what is phagocytosis?
When a phagocyte engulfs a foreign.
Right, a phagocyte will engulf a foreign bacterium, okay? So, what helps avoid are these things called capsules, okay? These capsules help prevent that bacterial cell, okay, from being engulfed by a phagocyte. And it's made, these capsules, and you'll be doing capsule stains in lab, right? So there's special proteins and waxes, or sometimes a combination of both, okay? And what it is able to do is the leukocidins are also can be produced by these capsules. Leukocidins destroy neutrophils and macrophages, okay? So not every cell that makes leukocidins has a capsule, some don't, but just be aware, you've got capsules and leukocidins. It's a great way to avoid phagocytosis. Okay, this just kind of shows you how it can block phagocytosis. All right, number 4, invasion of host defenses. Okay, anybody have an idea what this little microbe is? You might remember it from Bio 1. They're not really blue, green, and purple, red, or whatever. Okay. Do you know what microbe this is? It's a protist. Causes malaria. Okay. All right, this is a trypanosome. How can you tell? Well, trypanosomes have a flagella, and they also have this undulating membrane. So this is one of those critters that should remember from Bio 1. Okay. This is really, as someone who's trying to treat lots of microbial diseases, this is scary to me, because let me show you what happens. Okay? Antigenic variation. What is an antigen? What? Anything that stimulates the immune system, right? That's an antigen. Okay? So when you hear antigenic variation, what are you seeing? You're seeing antigens diversify. They change. Okay? So here's what happens. And we see this with malaria too. Okay? So you've got this clone, okay? And it's got certain antigens on the surface of it. It's a single cellular molecule critter here, this Plasmodium, okay? And then the immune system is able to wipe it out. So those that survive, what do? They switch it up. They mutate. What doesn't kill you mutates and comes after you to kill you later, okay? So what does it? It changes the antigen. So now what? Our immune system's like, whoa, what happened here? I haven't seen this. And so it takes a while, and then it will get these numbers out of control. And the survivors, what happens to them? They mutate again. This is going on constantly. Okay? This is amazing that it has, because why? Life from their perspective, they just want to survive. So what do? They're going to keep switching it up. That's what antigenic variation means. Okay? So yeah, it really truly is a moving target for the immune system. Totally a moving target. Okay, so we go back to the list here. Production of extracellular enzymes. Okay, here's a bunch of enzymes that you should recognize from your disease card list. Okay? Extracellular production of enzymes. These enzymes that digest and destroy tissues cause what are called cytopathic effects, okay? And these are coagulases, keratinases. Oh, the funguses are brilliant at making keratinases, because where do you find keratin? Your skin and your nails, okay? Hyaluronidases, streptokinases, collagenases, mucinases, and hemolysins. When you hear hemolysin, what should you think? Breaking of red blood cells, okay? So, yeah, all of these enzymes that do horrible things to the host, okay? This is really what allows a lot of pathogens to break through the basement membranes, okay? So usually you have a blood clot that forms around pathogens. But then the pathogens make streptokinase, and they dissolve the clot, and then they're free. That's pretty sneaky, okay? Hyaluronidase, what does that do? It destroys the cell-to-cell contact that you have in deep tissue, and then they're able to invade, okay? Crazy little, this is war, right? This is absolute war. They're going to try everything to try and get into the host. Okay. Any questions on this one? Okay. Yeah. What was the M? Mucinases. So you take Mucinex, right, to break down mucus. Well, they have them too. Yeah. C? Which one? There's two C's. The second one. The second one is collagenases. So what is that going to break down? Collagen, right. Okay. There's a list, the introduction to your disease cards. Make sure that you take a look at the list. It's there too. Okay. So, production of exotoxins. Let's talk about some exotoxins here. Okay. Exotoxins. These are proteins that are synthesized by the cell and then secreted outside. And you see these with gram-positive microbes. Okay? And there's many types. Okay? Often they're pathogen specific. Okay? Pathogen, S, what did I have there? Pathogen / species specific, so that should be species. And they are what they're called heat labile, which means that if you heat up, the protein denatures, okay? And you've seen that, that's an experiment that we do in Bio 1, when we take enzymes and heat them up and then they stop working, okay? But a lot of proteins are like that. They're very heat sensitive, okay? All right. So what belongs here? These are toxoids. The toxoids are altered exotoxins, which induce the antibodies, okay? And then antibodies are antitoxoids, which are produced by your immune system to neutralize them. So you've got a war going on between the bacteria making these toxoids and your antibodies trying to neutralize them, okay? So, as long as this bacterial cell is healthy, it's going to be secreting these exotoxins, which will have effects on the host. And, okay, what is a bacteria that produces a toxin? Staph aureus, yeah. Is Staph aureus gram-positive or gram-negative? It's gram-positive, okay. All right, have you ever heard of Botox injections? What are they doing? They're actually injecting an exotoxin that comes from botulinum into your face to paralyze your muscles so you can't smile anymore, right? Makes the wrinkles go away. Are there side effects for this? Absolutely, okay? I know they've been on a big marketing thing. I see it a lot. It's like, I got Botox when I was 40, and I'm like, what the hell? Really? Why? Why are you doing this? Now, there's some medical reasons for getting Botox. It does seem to help with certain type of migraines. Sometimes if you get a dysphonia of your vocal cords, they can give toxin to kind of help paralyze them a little bit so they can be better regulated. There are medical reasons for getting, Botox. But to go get it shot here and here and here and here, and then people come and say, well, I had a reaction. This is my shocked face. Of course, you're taking this very potent bacterial toxin that in high enough numbers will kill you and making your muscles, paralyzing your muscles. Wow. Okay. Yeah, pretty amazing. Yeah. So, all right, let's do the endotoxins. Oh, I forgot, there's examples of exotoxins. Okay, the diphtheria exotoxin, it inhibits protein synthesis. Erythrotoxin is produced by Streptococcus pyogenes, and it is, you can see here, what is it? And then enterotoxin, what is enterotoxin? What system does that affect? Entero, when you see entero, that's your GI. Okay, all right. And one type is cholera, the Vibrio cholera. And then neurotoxins interfere with nerve transmission, botulinum and tetanus, all produced by Clostridium. Okay, so those are some really common exotoxins. Yeah. Do you know about cholera? Okay. Cholera is one of those diseases you never want to get in your population, especially with kids. It can kill kids in 24 hours. You get the runs so bad that you produce what are called rice water stools. It's not even brown. It's like you did a colon prep. I mean, it's just fluid coming out of you so fast. Yeah, and when we usually see cholera after there's been like flooding or a hurricane or something like that. We saw cases of cholera. It's all oral fecal, and if you can't get your child treated and they've got rice water stools, you've got about 24 hours before they die. They're electrolyte imbalance, they don't have a buffer, and so they can die very quickly. Cholera is no joke. And you read that about Jon Snow, right? In chapter 1, who got the cholera outbreak to one area of, London that was a water pump, and that's where it was coming from, and it killed so many people. So yeah, it's no joke. All right, it's not just your normal, oh, I'm gonna take some Pepto-Bismol and I'll be fine. No, it's not. Okay, number 7, endotoxin release. This is so cool. Not for the patient, but the biology is cool. Okay, lipid A is a component of all gram-negative cells, and it's found in the outer part of the cell wall. Okay, now this is very non-specific. It's not species specific. It's not heat labile. So you run a fever, they're not going to go away. Okay, and they're released by dead, DEAD pathogens. Okay, now let's see how this works. Here's a lipid A. Okay, you know about gram negatives. They've got a little bit of cell wall, and then they've got an LPS layer, and they've got that's where, here's the endotoxin. Okay, the microbe dies, and that endotoxin is released. Okay, if you've got a patient that's got a significant gram-negative infection, and you start treating with antibiotics, and they're like, oh, I'm starting to feel better, and then they crash, and they start having a fever and stuff like that, suspect an endotoxin. Okay, because as an antibiotic is working, and more of these are dying, those endotoxins are released. And if you get enough, it's serious. I mean, your patient can crash. Okay? What can happen? They get fever, aches, weakness, breathing, heart problems, and can go into sepsis. Okay? All right, so when you got a gram-negative infection, which are really common, right? They're respiratory, sinus, prostate, UTI kind of thing, and you start giving antibiotics and your patient crashes, it's usually due to excessive endotoxin being released. Did you know that? Isn't that cool? Scary, but it's really cool. Yeah. Okay. Yeah, as long as it's not you. Yeah. Yeah, so we've gone through all 7 of these. Okay, so let's just revisit the pathogenicity here. Oh, there's another table. I love tables. Exotoxins versus endotoxins, just so you can have it. Again, I'm not going to say, oh, reproduce a table you saw in the PowerPoints. No, I'd rather give you, when we start doing case studies, you can refer to these. So, all right, so we're almost there. We've talked about all of the cytopathic portals of exit. Okay, and again, it's not necessarily the same as a portal of entry. Okay. Any questions about this section of the disease process before we go to the disease stages? Good? Okay, let's go to disease stages. This looks like a very, very simple diagram at first, but let's talk about it, okay? Here's the incubation period. You get sick, you get better, it resolves, right? If it were so easy, okay? If it were so easy. Okay, incubation period. Do you have any signs or symptoms then? No, okay. How long can this last? About 7 hours to 20 years, depending on the infection, okay? So this could be a long incubation period, all right? Prodromal period. Signs and symptoms showing up now? Yeah, okay, what's the difference between signs and symptoms? Good, perfect. Okay, so, yeah, this is the part where you're like, I don't feel well. I feel like I'm coming down with something. Ugh. Okay, then we have the period of illness. Do you have signs and symptoms here? Yes, you've got signs and symptoms here. Okay, now, during this period of illness, if your immune system does not respond properly, you'll die. Okay? You don't really have anything. You can get some medication sometimes. What was the problem here? Well, a lot of times during this infection time with COVID, okay, people would die. How long does it take to get a good antibody response? About 21 days. If you've never seen a pathogen before, that patient could very easily die before that 21 days comes, okay? And that's what happened with a lot of early COVID cases, is they just died before, because COVID was a novel virus, which means it's brand new. Nobody had seen it before. Nobody's immune system had seen it unless they got SARS in the Middle East in 2003, okay? So there's no immunity, and we had 0 treatments, okay? So hopefully we do have treatments. That's what I hope for, okay? And then we start to get a little better. We go through this period of decline. Now this part right here is critical, because at this point during the period of decline, you can get a secondary infection. What is a secondary infection? What's a secondary infection? Okay, one that comes after. What's an example of a secondary infection? Okay, yeah, pneumonia is a really common secondary infection. You get some viral illness and then you get pneumonia when your immune system is totally depleted, okay? And then this period of convalescence while you're trying to repair everything that got damaged. And this could last from a day to years. As we're seeing with people that have long COVID, they're in this period of convalescence for a long time, okay? A long, long time, okay? So, any questions about the stages of disease? Okay, all right, kind of put it into words, and another little, picture, and keep in mind some of the incubation times. We said 7 hours, okay? Some of the food poisonings are within hours, okay? Leprosy, HIV, Creutzfeldt-Jakob disease, the prion disease, they're in 20 years, okay? So we have all sorts of incubation times. Yeah. Again, you don't have to know this, just get an idea of what, would be here. Okay, let's talk about the portals of exit. A portal of exit is how the infectious agents exit their host so they can infect other hosts. So how does it usually happen? With body fluids, respiratory droplets, secretions, semen, and what's my favorite F word? Fomite. That's right, fomites. Good. Okay. So this is kind of a sampling of different terms associated with diseases. Okay? All right. For example, when you say something is localized, what does that mean? It's in one area, usually around the portal of entry. Okay? When you see systemic, what does that mean?
One system.
Means it's complete okay, system-wide. When you see focal, the term focal, what does that mean? Focal means that it's reduced to a specific body part or area, okay? That's a focal infection, which means it's in a certain region, okay? What does acute mean? Short term, good. What is chronic? Long term, good. And we just talked about primary and secondary infections, okay? So let's go over what the suffixes are. When you see itis, what should you think of? Inflammation, right. Emia, that it means it's blood related, okay? What about osis or iasis? This is all signs and symptoms, okay? Related, okay? And then if you see oma, what does that mean? It can mean opening, or if you see it like carcinoma, it's a cancer. If it's adenoma, cancer, okay? So, yeah, there's a lot of suffixes, and anatomy really got you warmed up for those. So that's good. That's really good. All right, let's talk about epidemiology. We've got a few minutes left here. Okay? When we do epidemiology, we're talking about all the factors of disease, everything here. Okay? And it's even more than this. We've got to include everything when we're trying to figure out the patterns of diseases. Okay? So let's go through some. Actually, let's finish this part up, and let me give you a short quiz, you whizzy, just for yourself, to see how you're doing on these two things. Okay, so give me a moment, and you can get out. You're not going to turn this in. This is going to be for you. Okay. Wanted to see how you do here. These are just some questions, quick concept questions. Okay. Are you ready to go?
Nice.
No. Oh my goodness, excuse me. I gotta put that daytime in. Okay, here we go. All right, these are questions from chapter 1 and 13. We're almost done with 13. So, there we go. What do you think your answer is? Which of the following does not describe a fungus? Okay, you got it? All right, what's the answer? It's delta, right? It's not photosynthetic. How many got that right? Okay, a little bit, that's good. Scientific theory has little or no evidence to support it and can be described as a best guess. True or false? False, okay. Yep. Okay, organisms in the same family must also be in the same class. Ooh, the same family, domain kingdom. Okay, it is true. Okay, domains, organisms in the domain Archaea have more DNA sequence similarity to coli, humans, or Archaea have no sequence similarity to any other organism. Okay, you got an answer? Okay, what'd you say? It's A. They're in it. Remember, these are prokaryotes. They're just extremophiles. They live in an extreme thing. Okay. Normal resident microbes are absent from the. You got it? What'd you get? Yep, lungs is right. Good. Normal resident. Oh, I got that one. That's a double. Okay. If a nurse transfers a pathogen between patients without becoming infected herself, the nurse has acted as the D? No, passive. She's not asymptomatic. She's not sick without becoming infected herself. Yeah. Okay. Occurrence of Lyme disease is mainly in areas where certain species of ticks would live. It is defined as a, we haven't gotten to this section, but you should have read it for sure. What is it? Endemic. Okay, so if I were you, you have 1, 2, 3, 4, 5, 6, 7 slides left. All right, don't wait for me to go through them. Okay, this is something because guess what? I'm going to expect that you've gone through chapter 4 by the time we come to class next time. Yes?